Another great day to talk about pharma. This is Kyle Cibolado from Group 1 3VMB, and I'll be discussing cholinergic transmission, only here in Vet Pharma 311. Let's go! Our learning objectives along the discussion would be to understand the basic concept of cholinergic drugs and the enzymes related. Next is to know the general concept or flow of cholinergic transmission. To differentiate muscarinic and nicotinic receptors, and lastly is to enjoy during the discussion. It's always better to have fun while learning. The cholinergic drugs, or the drugs that are used for cholinergic transmission, are defined as a category of pharmaceutical agents that act upon as the principal neurotransmitter in the parasympathetic nervous system, which is the acetylcholine or ACH. So parasympathetic nervous system are those that which is responsible for rest and digest response, which is also known as calming or cholinergic. Thereon, in cholinergic drugs, their common functions of what they do is that they stimulate the activation of parasympathetic nervous system by activating receptors for the ligand or neurotransmitter acetylcholine. That's why they are also called parasympathomimetic, because they mimic the effects of the parasympathetic nervous system transmitter. It is also often contrasted with noradrenergic or adrenergic drugs, since they oppose or inhibit the functions of cholinergic drugs, which is also explained in the noradrenergic or adrenergic transmission that will be further discussed on some other video. Also, cholinergic drugs will be also discussed on some other videos, especially in the topic pharmacology of cholinergic system. Now to the main topic, cholinergic transmission. It is defined as the process of synaptic transmission which uses mainly acetylcholine as a transmitter. Also, in the cholinergic transmission, the flow of the processes is the following: synthesis, storage, and release respectively. As mentioned previously, they are primarily found in parasympathetic nervous system. However, they are also in relation to sympathetic in a way that it stimulates for the skeletal muscle contraction. It induces sweating and others. Thereon, the sites where acetylcholine is a transmitter include autonomic ganglia, both sympathetic and parasympathetic. Next is the parasympathetic postganglionic nerve terminals, in which postganglionic is synonymous to postsynaptic, which means to run from a ganglion to an effector organ. Then the sympathetic postganglionic nerve terminals to sweat glands in man and cats. Then the nerve supply to adrenal medulla. Also the central synapses, or the CNS, both brain and spinal cord. Lastly is the somatic motor nerve endings and neuromuscular junction, or MEP. After this slide, as I discuss the processes, pay attention to the arrows so that you will be able to keep on track and will not be lost to know which part I am discussing about. Also, remember that acetylcholine is the protagonist in this discussion. To start with, we define the different areas in this figure. The axon terminal is in this area, shown by the purple bracket, which is also known as preganglionic area, where the acetylcholine synthesis occur. Meanwhile, this is the synaptic cleft, wherein it is the tiny space between two nerve cells across, where the neurotransmitter acetylcholine is broken into two. Next, in this lower area, it is the postganglionic cell, or synonymous to postsynaptic, where the acetylcholine runs to the effector organ. So, before I explain how acetylcholine is synthesized, I first start to explain how the formation of acetylcholine occurs, then proceeds to its synthesis. It starts with the acetic acid or acetate, which is taken from the drug or breakdown by the acetylcholinesterase of acetylcholine into two parts, acetate and choline. It penetrates the cell membrane through diffusion, and now that the acetic acid is in the cytoplasm through diffusion, it is first added with ATP, then results to acetyladenylate or acetyl AMP by the enzyme acetate thiokinase. Consecutively, the acetyl AMP is added with coenzyme A or CoA, then results to acetyl coenzyme A through the enzyme transacetylase, which means that from the coenzyme A, it delivers the acetyl AMP in the mitochondrion to be oxidized for energy production by the citric acid cycle or Krebs cycle. Then, it results to the product of acetyl coenzyme A. Then finally, the acetyl coenzyme A is added with choline, which results to acetylcholine and coenzyme A by the enzyme acetyltransferase. So where did choline came from? Choline came from here as a result from when the acetylcholinesterase breaks the acetylcholine into two parts, acetate and choline. It was then transported back into the presynaptic or preganglionic neuron by a high-affinity choline transporter or HACHT or CHT. However, obstacles can occur when hemicholineum and triethylcholine can competitively block choline uptake into the nerve and thus interferes with synthesis of acetylcholine. Now that acetylcholine is formed and synthesized, where does storage occurs? After synthesis in the axoplasm, the acetylcholine is transported via carrier mediated to the synaptic vesicles and stored until it is required to be released. However, along the processing of storage, an obstacle can occur when the vesamicle can competitively inhibits the uptake of acetylcholine into the vesicles. Now that the acetylcholine is stored, how is it released? When the action potential or AP, which is the waveform that controls the opening of voltage-gated calcium channels and contributes to the driving force for calcium ion, so again when the AP reaches the nerve terminal, the calcium ion enter the nerve terminal and cause contraction of the filaments connecting the synaptic vesicular membrane to the nerve membrane, thus pulling the synaptic vesicle in close proximity to the nerve terminal membrane and fusion of both vesicle membrane and nerve membrane. The presynaptic release of acetylcholine at autonomic neuroeffector junction, it is controlled by acetylcholine itself released from the same nerve terminal, which is the autoinhibition, and NOR-A is released from sympathetic nerves. An example of it, which is the homotropic and heterotropic presynaptic interactions respectively. Then, acetylcholine comes out of the nerve terminal or prejunctional membrane by exocytosis to the synaptic cleft. However, an obstacle can occur when the botulinum toxin and beta-bungarotoxin can block acetylcholine release at neuromuscular junction and thus cause paralysis of respiratory muscles, wherein the black widow spider toxin and ciguatoxin, which can cause an initial increase followed by blockade of acetylcholine release. So, how is acetylcholine received by the postganglionic cell? Now that the neurotransmitter acetylcholine is released, it is first welcomed by the receptors of the presynaptic cell, or it combines with specific post-junctional receptors called cholinergic receptor, which is another nerve or effector cell. And it is divided into two, muscarinic and nicotinic receptors. The different types of cholinergic receptors will be further discussed later on. So what happens after it is received by the receptors of the postganglionic cells? Now that after serving transmitter function or reacting with postjunctional receptors and initiation of postjunctional activity, acetylcholine is instantly destroyed in a flash, like suddenness by the action of an enzyme called acetylcholinesterase or ACHE. As mentioned before, acetylcholinesterase splits or hydrolyzes the acetylcholine into two parts, choline and acetate or acetic acid. Then the cycle repeats itself. Now that we described the general flow of concept on how synthesis, storage, and release works, more additional information will be discussed, such as getting to know the cholinesterase more and the two different types of receptors under cholinergic transmission, which is muscarinic and nicotinic receptors. So, acetylcholinesterase is called as true cholinesterase since acetylcholine is an enzyme found or present in the brain and erythrocytes of many species. Thereon, cholinesterase is defined as enzyme that hydrolyzes esters of choline. Butyryl CHE or BCHE or pseudo-CHE is another type of cholinesterase. Both the enzymes mentioned can be inhibited by anticholinesterase agents such as organic phosphate, insecticides whose toxicity is characterized by excessive accumulation of acetylcholine. So what is acetylcholinesterase? As mentioned on the flow of the cholinergic transmission, it's an enzyme that functions to hydrolyze acetylcholine or breaks them into two, which is choline and acetate or acetic acid. They are usually found at postsynaptic neuromuscular junctions, and they are necessary to allow a cholinergic neuron to return to its resting state after activation. In addition, it is also present in RBC membrane where its exact function is unknown, but it may regulate erythrocyte permeability. They also hydrolyzes acetylcholine at a faster rate than other choline esters including methacholine but not benzylcholine or butyrylcholine. Moreover, the pharmacological effects of drugs like neostigmine and physostigmine are due to inhibition of acetylcholinesterase, the indirectly acting cholinergic. On the other hand, butyrylcholinesterase are a non-specific esterase mainly present in blood plasma, live, and other organs. It rapidly hydrolyzes butyrylcholine and benzylcholine but slowly in acetylcholine, but not in methacholine. It hydrolyzes other esters such as procaine, succinylmethonium, and propanidine. Next, we discuss what are the different types of cholinergic receptors, what are its subtype, where can it be found, what are its agonists and antagonists. So the first type of receptor that we will be mentioning under the cholinergic receptors is the muscarinic receptors. They are named after muscarin, an alkaloid of mushroom Amanita muscaria. So why are they named after muscarin? It is due to their increased sensitivity to muscarin, a component found in mushroom, and they are lesser sensitive to nicotine. But they are also an acetylcholine receptors that form G-protein couple receptors, complexes in the cell membranes of certain neurons and other cells. So the following are the subtypes of muscarinic receptors M1, M2, M3, wherein M1 it is can be found where its location can be found at CNS, glands or gastric salivary and more, and autonomic ganglia, where its agonists are acetylcholine, carbachol, oxytremorin, and MCNA343. In antagonist for the subtype M1, it is the atropine, dicycloverine, ipratropium, tolterodine, oxybutynin, and so on and so forth. Meanwhile, in the subtype M2, it can be found at CNS, heart, and GIT. So its agonist is same as for the M1, and in addition, it is the methacholine. So its antagonist also includes atropine, the cycloverine, tolterodine, and so forth and so on. Next is in the subtype M3. Its location is exocrine glands, which are the gastric, salivary, and more. It also can be found in smooth muscles such as GIT, eye, and blood vessels. It does also have agonist same as for the M1, but in addition, there is bethanicol. Its antagonists also include atropine, dicycloverine, tolterodine, and so on and so forth. Meanwhile, in the subtype M4, it can be located at CNS and lung. So its agonists are also the same as for the M1, and its antagonist also includes atropine, dicycloverine, ipratropium, tolterodine, and so on and so forth. Same as the M5, its agonist is same as for the M1. However, in its location, it includes the CNS, salivary glands, and eye, which is the iris and ciliary muscles. Its antagonist includes atropine, dicycloverine, tolterodine, oxybutynin, and ipratropium. Now, to the second type of cholinergic receptors, it is the nicotinic receptors. They are named after nicotine or alkaloid of leaves of Nicotiana tabacum. Since unlike muscarinic receptors, nicotinic receptors does not stimulate muscarinic acetylcholine receptors, but selectively binds to the nicotinic receptors instead. Thereon, they are ligand-gated ion channels, activation of which results in depolarization and excitation, wherein it is a channel protein that upon binding by acetylcholine, it opens to allow diffusion of cations. So under the subtypes of nicotinic receptors include the skeletal muscles and ganglion, wherein in the skeletal muscles, they can be found at neuromuscular junction or postsynaptic, wherein its agonists are the acetylcholine, carbachol, succinylmethonium, PTMA, and its antagonists are there mentioned. And meanwhile, in the subtype ganglion, it's located in the autonomic ganglia, which is in the postganglionic. Its agonists are the acetylcholine, carbachol, nicotine, epibatidine, and the MPP. Its agonists include mecamylamine and trimethaphan. In addition, it also has subtype CNS, which is the A and B. They are both located in the brain, pre- and postsynaptic, wherein the agonist of the CNS-A is the nicotine, epibatidine, acetylcholine, and cytosine, where its antagonists are mentioned on the table presented on the slide. Next, in the subtype B, its agonist is the epibatidine and the MPP, wherein its agonist is also mentioned on the slide. Feel free to pause and check the table presented on the slides. So that's all for now. I hope you learned something in this video. Pharmacia!